Hello everyone. I'm Eduard Ebberink. I'm a postdoc in the lab of Albert Heck at Utrecht University. And in this webinar, I will discuss some of the work I did on adeno-associated viruses and characterizing them with mass photometry. And then in particular, look at the capsid stability upon heating the AAVs. So first, a short introduction. AAVs, adeno-associated viruses, they kind of became the vehicle of choice within the field of gene therapy. And on this slide, I've put up the therapies that have up till now been approved either by the FDA or the EMA that are based on AAVs. And I've made this slide actually some time ago, and I have to keep adding new therapies because more and more are becoming approved and available. With the most recent one here in the corner, and this one here in 2024, which is directed also against hemophilia B, is being the latest addition. Of course, there are many more trials ongoing and research into the use of AAVs for gene therapy. So hopefully in the future, I can keep adding more and more therapies to this list, which would, of course, be very exciting to see. So AAVs, they're very small protein capsids and can have a genome of single-strand DNA of about 5 kilobases. And the total size would be something about 25 nanometers. And why are they now so popular as a vehicle for gene therapy? Well, AAVs, they're known to induce only a very low immunogenic response when given to people. And the AAVs in nature are known to be non-pathogenic, so they cannot replicate themselves. They need factors from other viruses in order for them to become to be able to replicate in a cell. So next to that, if you have a recombinant AAV made and have a gene of interest that you want to express in a certain cell, then if a cell is transduced, it actually has quite a long-term expression. And there are many different variants of AAVs. Well known are the serotypes, but there are beyond that even many more other variants. And they all have a certain cell type that they can transduce. So it's quite a broad tropism for these AAVs. And of course, there's also still research ongoing into developing more capsids and more different variants of AAVs in order to really specifically target certain cell types. All right, so why was our lab actually interested in AAVs to begin with? So if you would have a pool of AAVs made from cells, insect cells or mammalian cells, such as HEK cells, then of course you want to know after purifying these AAVs, how good are they? How many of these AAVs actually contain a genome with the gene of your choice? Because if you would create a pool of AAVs, recombinantly, only about 5 to 30% of these AAVs actually contain a genome. The rest is basically empty. And there are already quite some techniques available in order to characterize this, to see how many AAVs of your pool have actually a genome. They are based, for instance, on PCR or on ELISA or electron microscopy. And kind of the gold standard, I believe, and which is often used, is ultracentrifugation. And these techniques all have their advantages and disadvantages, but we are a group that is mainly focused on mass spectrometry, and we thought, okay, can we also develop a method based on mass spectrometry in order to answer this question of how many AAVs actually are filled and how many are empty. So basically, what we would do is our approach would have been to determine the molecular weight of each individual AAV and say, okay, this one is filled and this one is empty. And indeed, in the past, within our group, a method was developed where we were able to use a mass spectrometer, in our case, an Orbitrap-based mass spectrometer, where we can analyze for each individual AAV the mass. And basically, what we would do is allow only a few particles or even a single particle at a time in the mass analyzer in the Orbitrap and detect the charge, in this case on our capsids, and based on that extract the mass, and we could construct a mass histogram as shown here. So for this particular pool of AAVs, we would see that the majority of the capsids have a mass that corresponds to containing a single genome. But we can also see that some capsid actually has a mass that's a little bit higher than that, so they would have extra material packaged. And we can also see that there are still a few empty capsids in this pool of AAVs as well. So while setting up this method, a new method came about, which also caught our interest, but is based on a completely different principle, which is mass photometry. So I will go a little bit more into detail on how to use mass photometry to measure AAVs later. But here we principle do the same, that we can detect single particles, single AAVs, and determine the mass for each AAV, and then build again these mass histograms. So it's the same pool as before, and we see again a peak of AAVs containing just single genome, some that have extra material, and again some empties. And both of these techniques we can nicely use in order to answer this question, like how does our pool of AAVs look, like how many of them actually contain a transgene, how many of them are empty, how many of them actually have more than just a single genome or have half of a single genome. So both techniques appear to be in good agreement with each other, so we can also use them nicely in order to characterize the AAVs. And if you're interested, there's also a webinar about this available still online, I believe, about comparing these two techniques and measuring the AAVs. But of course, we were also interested in what would be the next step. What could we do more with these techniques and characterize our AAVs even better? And one thing we were interested in as well is to look at the stability of the capsids of the AAVs, for instance, upon long-term storage or when you incubate them at different temperatures. So what would eventually happen to these AAVs? Can they form aggregates, or are capsids being lost? Filled capsids, are they releasing their genomes so that you end up with empty capsids, or do they simply break either with the genome still attached or also released? And can we use our single molecule approach techniques such as CDMS and mass photometry in order to investigate the fate of the AAVs and the stability of the capsids? So in the past, research has been done, of course, on determining the stability of AAV capsids, and what usually has been done is incubating the capsids at higher temperatures, for instance 65 degrees or even higher, and see what happens to the AAVs. For instance, some examples that have been done, people have used differential scanning fluorescence, so DSF, and thereby basically you have your AAVs in solution and you add a fluorescent dye and you increase the temperature, and at some point the capsids will melt and the capsid proteins will denature, and then the fluorescent dye can bind and the fluorescence increases a lot, and this you can measure. And interestingly, the point of melting, denaturing of the capsid proteins is really specific for AAV variants for different serotypes. You can actually use this technique also to identify different AAV serotypes. However, this gives more information about the actual denaturing of the capsid proteins and not necessarily the different stages of capsid disassembly. Another technique that has been used as well is immunostaining, so using antibodies. You have, for instance, antibodies that can bind just a full intact capsid, and you have different antibodies that can bind parts of the protein that becomes exposed upon capsid disassembly. And you can use, for instance, Western blotting or dot blotting and increase the temperature of your AAVs and then see if the antibodies still bind. So you can actually see that if you increase the temperature at some point, the antibody that can only bind to a fully intact capsid will stop binding because the capsids fall apart, while others start binding because new parts are becoming exposed that can interact with these antibodies. So this is also a very nice technique, and you can really follow the disassembly of the capsid. However, Western blotting, dot blotting might not be that suitable for high throughput way of working. Also being used, and which works very nicely, is atomic force microscopy, so AFM, because there you can really see what happens to your capsids after incubation at a high temperature. So for instance, in this case, you can really see that at some point, the genome of the AAVs is being released, and you can detect these genomes as well. And even if you go at a higher temperature, here at 75 degrees, then there's no capsid anymore visible. It's just the released transgenes that you can see. Using this, you can really follow the stages of capsid disassembly. However, again, may not be that suitable for a high throughput way of working. And also perhaps you need some knowledge about working with AFM in order to do this. So how about the single molecule techniques that we can use in our lab? Can we also apply them in this context of AAVs that have been heated and see what happens to the capsids? Well, let's first start with charge detection mass spectrometry, CDMS. So already other groups have been quite active as well on this, using mass spectrometry and also Orbitrap based CDMS in order to characterize the AAVs that have been warmed up. So for instance, in this experiment, there they've very nicely used a pool of AAVs and incubated them at 45 degrees, as you can see here. So we have some filled particles, we have overfilled particles, and a few empties. And they kept this solution at 45 degrees, sprayed the AAVs in the mass spectrometer, and while the solution is being warmed up, I keep measuring the AAVs. And as you can see at 50 degrees here, already some filled particles and also overfilled particles start to decrease, while at the same time, the amount of empty particles that they can detect starts to increase. And this becomes more and more clear if you incubate it at higher temperatures, so 55 degrees and even at 60 degrees here. You can see that the overfilled particles are now basically completely gone. There's still a few filled particles left, but most of them are now empty particles that they can detect using the mass spectrometer and using charge detection mass spectrometry. Also in our lab, we tried some experiments. In this case, we used a different set of AAVs, as you can see here. So we kept it at room temperature at first. You can see here the amount of AAVs that have a single genome. And also here there are a few empty particles. But if you now keep the same temperature, 65 degrees, and incubate at different times and measure then the AAVs, you can see also here that even after incubating at 2.5 minutes, at 65 degrees, there's a huge decrease in the amount of filled particles, while we now can detect mainly a group of empty AAVs. And at 10 minutes, this is even more clear. The filled particles are basically completely gone, and we detect an enormous amount of empty particles. So both of these experiments and using charge detection mass spectrometry, it really supports the idea that what we see is empty or filled capsids that release their genome, that eject their genome, and that we then are left behind with empty capsids that we start detecting. So CDMS really works well. However, it might be slightly tricky to properly quantify the AAVs because, of course, it depends on how you set the mass spectrometer, which settings you apply, and can really affect the amount of particles that you can quantify of the filled and or the empty AAVs. So that still requires some knowledge, some expertise. Okay, then how about mass photometry? It's really easy to use. It's in solution and can be very quick. So we're really interested in, can we apply mass photometry on our heated AAVs? So how do we measure our AAVs with mass photometry? So mass photometry is a microscopy-based technique, and we use a SAMUX mass photometer in our case to measure the AAVs. And what happens there is that we have a glass slide, and a laser light is shined on below here of the glass surface. And most of this light is reflected actually by the glass surface. But if an AAV lands on top of the glass, it also scatters a bit of the laser light, and the scattering will interfere with the reflected laser light that you can measure as a bit of contrast. So these typically black dots basically that you can see here are a change in contrast upon landing of an AAV on top of the glass surface. And the important thing is that the amount of contrast that you measure is scaling with the mass of the particle. So what you could do is create, for instance, a standard curve, a protein standard using thyroglobulin, which has several multimers, so a monomer of 335 kilodalton, a dimer of 670 kilodalton, a trimer and tetramer. And you can basically measure the contrast values of these different multimers and create a standard curve like this. And then you know, okay, well, I have a certain factor for my contrast value, and I can extract the masses from my measurement. And for instance, in the case here that we have AAVs measured, we can see, okay, we have a sample of empty AAVs with a mass of about 3.8 megadalton. Okay, so let's see how this looks if we use mass photometry while heating up our AAVs. So we have here a pool of AAV8 with a genome that can express GFP. It's about 1 megadalton in size. So a filled AAV particle will be about 4.9 megadalton. And in this particular pool of AAVs, there are also some empty capsids present. And we're going to warm this sample up at 65 degrees and then see what happens. So to start with, we incubated just at 37 degrees for about 15 minutes. And we can see here our mass photometry measurement. So we have our filled particles here, and then indeed some empties also there. If we then calculate the percentage of filled particles, it's about 90% or so. So this is from several repeats of this measurement. Now, if we incubate for 5 minutes at 65 degrees, we see a drop in the amount of filled particles, which are now more close to the number of empty particles. And we also see a new peak emerging here at about 1 megadalton, which presumably is the released genome. And also, if we look at the percentage filled, it starts to drop at about 60%. If we incubate even longer, at 65 degrees, we even see a further drop in the amount of filled particles, which is now below the number of empty particles. And again, this peak is still here at about 1 megadalton for our released genomes. And also in the filled percentage filled, we see this drop now at about 40%. So this really supports the idea that upon heating of the capsids, the genome is released and we end up with empty particles in the end. Much as we saw earlier with CDMS, but also with AFM. So here another AFM image of another experiment where AAVs were incubated at 65 degrees. And indeed, also here we see genomes that are released and then still some capsids that are present. However, if we look at the total number of AAVs that we measure following heating, also that dramatically drops. So here we see the total amount of AAV that we count in a measurement. And after heating the sample, the number of AAVs that we detect drops dramatically after 5 minutes, but also after 15 minutes, it even drops a little bit further. So it could also be that alternatively, the genome is not released by the filled capsids, but instead the filled particles just disassemble and disappear. And the empty particles that we measure here are actually the particles that were already there at the start, but we now measure them more clearly because they are more heat stable, for instance, and do not disassemble as quickly as the filled particles upon heat. So now we have two potential mechanisms of what could happen with our AAVs upon heating. One is that during heating, the filled AAVs eject their genomes and create newly formed empty capsids. But on the other hand, what also could happen is that the genomes are released, but the capsids completely disassemble. And the empty capsids that we measure now are actually the ones that were already there, but these are much more heat stable. And that's why we see them better during the measurement. So in order to find an answer to this question of what's really happening with our AAVs upon heating, we actually wanted to use an internal standard that is not affected by heat or by anything else during the measurement, and that gives us a better handle on the quantification of our AAVs during these mass photometry measurements. So ideally, such a standard should, of course, be stable in an aqueous solution. It should also be able to tolerate heat, so high temperatures. And also important, the mass should be falling into a range where it's not interfering with anything else of what we measure. So for instance, our AAVs, but also not of the potentially released genomes and perhaps fragments of the parts of the capsid. So ideally that should be in a mass range around 2 megadalton. Well, at first we tried some proteins that we thought would be heat stable and also nanoparticles, but that didn't really work out. We quickly turned our attention to DNA that we thought would be heat resistant, and then especially the DNA plasmid of PBr 322, which also has a nice mass. And when we started to measure at first this DNA plasmid, we end up with our first challenge here, because DNA is quite negatively charged and the glass surface has these hydroxyl groups and they don't really interact with each other. So what we end up with is a measurement where you hardly see any landing events of our DNA plasmid. So we had to modify our glass surface first, and that we did with AAVs. So we coded our slides with AAVs, and now we introduce these amine groups, and then we can indeed see again our DNA plasmids start to land on the glass surface, and we can pick up the signal. However, what you might also see is that some of these signals have a bit of a strange oval-like shape, which we did not see earlier, which is different from our circular shapes, which we are used to with our mass photometry measurements. So here we have a frame with a zoom, and you can clearly see these strange oval shapes, which are different from the circular landing events that we are used to see. And this would be because the DNA plasmid is quite big, it has a quite long length, and it now starts to become close to the diffraction limit of the mass photometer. And we basically distort the sample a bit. We can see these strange oval shape landing events. So we also wanted to analyze it a little bit further. So we take our frames and then basically look at every landing event and see if we can fit an ellipse. And most of the times we see these nicely circular landing events, but sometimes indeed we can fit an ellipse as well. And we were afraid, does this also interfere with our actual measurement? Well, luckily not. So here we have a histogram of all the contrast values of our measurement. And if we have the normal conventional settings of the mass photometer software, it has a quite strict filtering, and it doesn't pick up any of these strange oval-shaped landing events. So we have here a nice Gaussian curve of all the contrast values that we can pick up. And if we get rid of this filtering setting, you start to pick up these oval-shaped events as well, which add also lower contrast values. But if you have the normal settings, then we don't pick up these weird oval shapes. So that is nice. And then, of course, we wanted to measure our DNA plasmid and heat it up as well. So here we have a measurement of our plasmid and then analyzed also for the mass value. And it's about 1.9 megadalton, which is not really what we expect on the theoretical value, which is about 2.8. But this is because we use a protein-based thyroglobulin standard. And the contrast values of protein is different than those from DNA. So you would need a specific DNA-based standard curve in order to get the proper mass. But we use the thyroglobulin, so the mass is now about 1.9 megadalton. But that's actually nice because it then fits nicely within our measurement compared to the other masses that we see. And if we heat up the sample, this is just DNA, for 50 minutes, 65 degrees, and we measure it again, we don't see much difference. So that's nice. It really seems to be stable against heat. So, but if we now repeat the measurement using the same AAVs as we saw earlier, so AAV8 containing a genome expressing GFP, and in this mix there are also some empty capsids that we can see. And I forgot to mention these were made in HEK cells. And what if we take this sample, again measured at 65 degrees, but this time we've spiked in our DNA plasmid. All right, so first we start with just a measurement at room temperature. So here we have our filled particles, we have our empty particles, but this time we can also detect our plasmid DNA. But what if we take the sample and heat it up at 65 degrees for 15 minutes and then again measure? You see a huge drop again in the amount of filled particles, but maybe also a drop in the amount of empty particles. And we see our DNA, which do not really change much. So using the DNA signal, we can actually normalize the measurement, and we can really look at the fold change of the empty and filled particles before and after heating the sample up. So here we have just our measurement before heating it up, being done at room temperature. And if we now incubate it at 65 degrees, we see this about 40-fold drop in the amount of filled particles. But also the empty particles, we might see a small drop, about a 3-fold or so, in the number of empty particles that we can detect. So this, of course, we can do at different temperatures. So I just showed here the one where we incubated at 65 degrees, but we can also do it at 60 degrees and 55 degrees. And here on top, we have the measurement done before heating. So just incubating it at room temperature. And then after heating, we repeat the measurement. And we can clearly see that at 60 degrees, indeed, again, a drop in the amount of filled particles. And at 55 degrees, maybe there we see that not much is happening. That maybe is a slight drop, but not that much. And we can actually calculate again for all these measurements the fold change that we see in the amount of filled particles and the amount of empty particles, and plot it in a graph like this. So here we have the different temperatures that we measure, and then look at the fold change of either our empty particles or our filled particles. And the filled ones, you really see that it changes much more dramatically with increased temperature, while the empty particles remain a little bit more stable, perhaps. At least we do not see a huge increase or accumulation of empty capsids that we might thought to see if the genomes are just released upon heating from the filled capsids, and we end up with creating more and more empty particles, which does not seem to be the case. Okay, so what does seem to happen, interestingly enough, is that the empty particles that were already there in the sample before starting up the heating of the AAVs, they do seem to be more heat resistant than the filled particles. So although we do not see any new antiparticles being created as filled particles eject their genome, we do see that the empty particles that were already there from the beginning, they appear to be more heat stable. So we also wanted to measure another sample, again AAV8, also made from HEK cells, but this time they were prepared in absence of any transfer vector. So there's no DNA within the capsids. They were really made on purpose to be empty capsids. And also those we then heated up and measure. So what we can see here is we have, again, our measurements, and at the top where the sample is incubated at room temperature. So in this case here we have our empty AAVs and our DNA plasmid. And then following a measurement after room temperature, we incubated at higher degrees, for instance, here at 60 degrees, and measure again. And we see here our empties and our DNA, and nothing changes really. So the empty AAVs, they seem to be stable. If we incubate it at a higher temperature, also there we see that the empties are again still there, and they seem to be fine and stable. But if we now increase the temperature a little bit more, so 68 degrees, then all of a sudden we see that compared to the measurement previously done at room temperature, the amount of empties completely decreases, collapses basically. The DNA still is fine, but our empty AAV is really gone. They really decreased tremendously. And because we have our DNA, we can again look at the fold change over these different temperatures and plot them together with the previous measurement. So here on red, we now see our new sample of the empty AAVs, and up to 65 degrees, not much is happening to the AAVs. They seem to be fine. But if I go up slightly at 68 degrees, then there's a huge decrease in the amount of empty AAVs that we can measure. Okay, so it appears that the empty capsids are indeed much more stable than their filled counterparts. And we wanted to see if this also applies to, for instance, different serotypes. So we also have a sample of an AAV9, which has more of an equal mix of filled and empty particles. And the genome also here expresses for a fluorescent protein, which is in size about 1.2 megadalton, the whole genome. And we want to see that also here we can observe that the empty particles are much more stable. And in this case, the empty particles are actually formed as a side product for during the expression and packaging of the filled AAVs. So how does this look like? Well, we again did our measurement at different degrees. So on top here are all the times that we first incubated at room temperature, and then below, after heating up the sample, we measure the same sample again. So at 55 degrees, we see that we have here our empty particles, and here are filled particles, and not that much is happening to both of these filled or empty particles. But if we incubate it at 60 degrees, now we see that our filled particles are disappearing or decreasing, while our empty particles stay stable. And at 65 degrees, this is even more clear. So the filled particles are really completely gone, while the empty particles are much more stable and remain within the sample. Again, using our DNA here that we also measure, we can look at the fold change of our empty and filled particles before and after heating. And we can see that indeed the empty particles, they stay quite stable, while the filled particles at some point starts to decrease much more in. So I think we can clearly see now and clearly state that the empty capsids are more heat stable than filled particles. So on the basis of the measurements we just did on filled AAV8 and empty AAV8, and the sample with a mix of filled and empty AAV9 capsids, I think we can say that the filled capsids indeed start to release their genome upon heating, but then probably disassemble and are lost and do not accumulate in new empty capsids. And on the other hand, empty capsids that were already there in the mix, they are more heat stable than the filled capsids, so they persist throughout the measurement. So far, we've mostly looked into the difference between the empty and the filled capsids and their stability upon heating, the AAVs, but it would of course also be interesting to look in the difference between the different serotypes. So here we have a sample of AAV2, which actually has an identical genome as the previously measured AAV8. So they both are made in hexels. They both have a GFP genome, which is about 1 megadalton in size. However, the capsid exterior is different in this case. It's an AAV2, and we can compare it to our AAV8. So let's see. So this is the previously measured AAV8 again. This time depicted slightly different. Here I overlaid the different mass photometry measurements. So in darker gray, it's the measurement before heating, where they were incubated at room temperature, and then in light gray, the measurement after heating the sample at the elevated temperatures. And here in the corner again, the quantification with the fold change of the different empty and filled particles. So how about AAV2? Well, so if we look here, you can see at 55 degrees incubation, either heating the sample at 55 degrees or keeping them at room temperature, not much is happening. Here we see the filled particles, we have our empty particles here, and also a few of the partially filled, and not much is occurring between these different AAVs. If we incubate though at 60 degrees, we do see quite a big difference. So now the filled particles are almost all lost, and we still have some empty particles left. And if we even go higher, so 65 degrees, again, filled particles are gone. But also now we can see maybe a decrease in our empty particles as well. So if we look at the fold change here, the quantification, then we see that indeed at 55 degrees, not much is happening. There might be even a slight increase in empties. But at 60 degrees, there's a huge drop in the amount of filled particles and also the partially filled, and then even the empty particles, they stay stable, as we saw earlier. But if we now go at 65 degrees, we also see that the empty particles start to decrease together with the filled particles and the partially filled, which are really almost completely gone. So something particular about this AAV sample, the AAV2, is that we also observed quite some aggregates landing on the slide. So what you would see, for instance, in this video shown here, is that these aggregates kind of float over the slide and at some point really attach on the slide and give this huge signal of contrast that we can see. And basically we counted the amount of aggregates that were visible during our measurement, and we saw that for this AAV2 sample, there are quite some aggregates that we could detect. Much more than in the sample measured previously, the AAV8. There we occasionally see an aggregate, and also only when the sample was heated. But in this case, we see also for the AAV2s that at room temperature, we see quite some aggregates as well. And why do we see them so well now? Could also be perhaps that we use optical slides and instead of glass, normal glass slides, that we can see them so nicely here. Unfortunately, mass spectrometry maybe is not the best method to quantify the aggregates from AAVs. There are better methods available, and very nicely shown in this paper of this group is that they used dynamic light scattering and also warming up of the AAVs, and then see how they could quantify the amount of aggregates that were there. So how about even a completely different sample? This is again an AAV8, but this time made from insect cells. And also here the genome is slightly different. It's again for GFP, but a little bit shorter, a smaller genome. And we have filled capsids here, but also a small amount of overfilled particles as well. Okay, so how does this look? Well, here we also show measurement done at 45 degrees. So we have here our filled particles. We have our overfilled now as well here, and a small amount of empties. And if you incubate it at 45 degrees or keep them at room temperature, not much is happening. But if you start to incubate them at 55 degrees, then we do see a huge decrease already for our filled particles and overfilled. And at 60 degrees, that's even more pronounced. So a drop in our filled amount of filled particles that we can measure, and also the overfilled. And of course, 65, that's even more clear. Again, using our DNA signal in each sample, we can quantify the amount of empty and filled and overfilled capsids while heating them up. And we can see here in our fold change plot that at 55 degrees now we see that the filled and overfilled particles indeed start to decrease, which continues during 60 degrees heating and 65 degrees, while the empty particles remain much more stable and persist throughout the measurements despite any increase in temperature. So what can we conclude from these measurements with the different capsids? Well, it's hard to make any general conclusions, because what we really see is that for each sample there are specific differences that we can pick up. For instance, at which temperature the filled particles start to disassemble, and also, for instance, how stable the empties are, and if we can see any aggregates, and if these aggregates only are visible when you heat the sample or not. These are all quite specific to the samples that we've measured so far. Okay, something interesting that we noticed during these last measurements is that upon heating, a new peak occurs, which has a mass which is about the same as the genome of the filled capsid. So here, for instance, for the AAV8, we could see this new peak emerging. And also in the measurements before, where we did not yet use a plasmid DNA and also just conventional glass slides, after the heating of the AAVs, we see this new peak, which has about a mass of 1 megadalton, which corresponds to the mass of the genome of the filled capsids. This was the case for the AAV8 from HEK cells, but also with the AAV8 from insect cells. We saw this new peak arising, which has in this case a mass about 0.7 megadalton, corresponding to the size of the genome. For AAV2, this was unfortunately not so clear. It was more tricky to detect, or we could not detect it. But for the AAV8, we clearly see this new peak, which presumably is the single-stranded DNA that's being released from the filled capsids after heating. So we wanted to really make sure that that's the case, and thought of a different assay where we would first heat our sample, but after heating, we would add a bit of DNase and see if that would digest the new peak here that emerged after heating, just heating of the AAVs. So in this case, we have our AAV8, we incubate at 65 degrees, we see a drop in the amount of filled particles, and we see this new peak here at 1 megadalton, which is presumably our single-stranded DNA. And then we add a bit of DNase, incubate at 37 degrees again, and measure with mass photometry. And we see that this peak indeed disappears. So indeed, it looks like the peak that we first saw was our released single-stranded DNA genome. So that's quite interesting, but what we also see at the same time, to our surprise, is that the amount of filled particles start to drop again just by adding DNase. So if we also repeat this experiment several times, every time we heat the sample, we have about 50% of filled particles and 50% empty. But if we add DNase on top of that, we see that this drops from 50% to about 30%. So this was interesting, and we wanted to make sure that what we saw was because of the DNase and not something else, because we also have to add a bit of magnesium to the DNase in order for it to be functional. And we know that divalent cations can also interact with the capsids and changes their stability. So we first did a control experiment where we again heated up our sample. So we have here 50% empty, 50% filled, and this peak here of a single-stranded DNA, and then added only a bit of magnesium and incubated that at 37 degrees and measure again. And then we see that the single-stranded DNA peak is still there. And also the amount of particles, empty or filled, doesn't really change much. But if you, on top of this, then add the DNase, then we see again a drop in the number of filled particles compared to the amount of empty particles. And also the single-stranded DNA peak is now again gone. So this was, of course, quite an interesting observation. This would mean that there is a population of AAVs which looks like they are properly filled and are okay, but in fact are not. They might be broken, or the DNA might be stuck on the outside, and adding of DNase would uncover this population of AAVs actually. So we wanted to check this out, but also for different temperatures. So here's the measurement again when we incubate our AAV at 65 degrees. So we start with 50% filled and 50% empty. We add DNase, we see a drop in the percentage filled, and the fold change in this drop of percentage filled, we plot here in the graph, so which is close to the twofold change. If we incubate the sample first at 55 degrees, we also see a small drop in percentage filled after adding DNase. But if you would first incubate the sample at 45 degrees or lower, then not much is happening. So how is this for our other AAV8 sample, the ones that were made from insect cells? Well, we performed the same experiment there. And also here we can see an effect of the DNase. So if we would first heat our sample at 65 degrees, and we look at the percentage filled that we have then, which is about 40%, we add DNase, incubate again at 37, and then measure. We see another drop in the amount of filled particles to about 20%. Again, about a twofold change. But in this case, also incubating the sample first at 55 degrees has a certain effect, and also at 45, and even incubating the sample at 37 degrees and then add DNase would give you a small drop in percentage of filled particles, even at this relatively low temperature. Okay, so how's it for the other serotypes that we have? So we have the AAV2 samples from HEK cells and also a sample of AAV9. How does that look? Well, If we do the experiment there, it actually looks a bit different. So this is our AAV2 sample. If we incubate this sample at a high temperature, so 65 degrees, we have hardly any capsids left. So the circles are very small. It means that there are only a few AAVs that we can count. So it's not really a useful sample. But if we incubate first at 55 degrees, we still have about 60% or so of filled capsids left. And if we add DNAs on top of this, then actually nothing changes, as we can also see here in the histograms of the mass photometry measurement. And also at the lower temperatures, nothing really happens. Even if you add DNAs or not, it doesn't change the amount of filled and empty capsids that we can measure. How is this for our AAV9 sample? Well, also there we see a similar thing happening. So if also this we incubate at a very high temperature, let's say 65 degrees, we only have a few capsids left, so we cannot really use this temperature. But at 55 degrees, if you incubate it for 50 minutes, we have about 30% or so of filled particles left. If you add DNAs on top of this, then it again doesn't really have an effect. Nothing really changes there. So what can we conclude on the basis of these experiments, where we have a combination of the heating of our AAVs and the addition of DNAs? Well, in some cases, we can clearly see the actual genome that has been released by the capsids that are disassembling upon heat. And we can also see that following heating of our AAVs, there's a population of AAVs that appears to be filled, but they might actually be broken, or at least the genome is exposed to the outside where DNAs can access it and starts to digest it. But also this appears to be quite a sample-specific effect. We do not see it for all our samples. All right, so what can we say now about all the measurements and all the results that we saw during this presentation? Well, one thing that keeps reoccurring is that some properties of the AAVs upon heating, such as the temperature as they start to deassemble, and whether or not we can see the released genome, whether or not we can see aggregates, and if they are dependent on temperature. These things appear to be really specific for the batch of AAVs or the type of AAVs you're measuring at that moment. But more in general, what we could see is that empty capsids appear to be more stable than filled capsids. And after heating, there can be a population of AAVs or filled AAVs, presumably filled AAVs, which have the same mass as a filled AAV, but in fact are actually broken, and the genome is accessible to the outside to, for instance, DNAs. And this can already happen actually at a quite low temperature, as we saw in one case. And then next to that, I would say that mass photometry is quite a convenient tool to try things out, to set up certain assays, to modify assays. So we can modify the slide, we can add reagents such as DNAs, and it's such a fast measurement, and it's done in solution that you have quite some options in order to try things out and to find answers on your question about AAVs and, for instance, capsid stability. Okay, with that, we come to the end of my presentation, and I would like to thank some of my colleagues, such as Victor, Evelyn, Arjan, Nadia of the native subgroup, and of course Albert for his supervision, my fellow colleagues from the BioMS group, and Helena and Irene from Sirion, now Revity, for supplying some of these nice AAVs, and Alisa, Marcus, and Marco from Roche for their help. And of course, you for listening to this webinar.